Hello everybody, I'm Konstantinos Partsinevelos from the University of Catania, and the title of my presentation is Vanillin flavor in e-liquids. Is it dangerous for endothelial cells? The Replica project. This work is a part of a larger replication study related to Coehar's Replica project. Coehar is the Center of Excellence for the Acceleration of Harm Reduction of the University of Catania. As is customary with the replica study, these experiments were independently conducted in 4 international laboratories: the University of Catania, Italy, the leading center; Temple University, USA; University of Kragujevac, Serbia; and University of Pajajaran, Indonesia, following standardized and harmonized SOPs. The work that we replicated is flavorings in tobacco products induce endothelial cell dysfunction by Fetterman and colleagues. The purpose of the study was to investigate the impact of vanillin, a very popular flavor used in e-liquids, on endothelial cell function, evaluating cytotoxicity, oxidative stress, and nitric oxide bioavailability. Fetterman used for the exposure a furnace, and the vanillin was added drop-wise into the heated area and aerosolized. In contrast, we used standardized methods, including real liquids, with the use of propylene glycol and vegetable glycerin, commercially available e-cigarettes, and ISO-compliant vaping machines in order to verify whether the results were reproducible and applicable to real vaping experiences. Fetterman diluted the vanillin in ethanol, and the aerosol collection was achieved using an ethanol solution in a glassy pitcher. To adhere closely to their experimental procedures, we also did the same. Fetterman evaluated cytotoxicity performing tunnel assay, which is not typically used for the direct evaluation of cytotoxicity, but is commonly employed for detecting DNA fragmentation and apoptotic cell death. Instead, we performed NRU assay, which provides information about overall cell damage, regardless of the specific mechanism of cell death. For the evaluation of oxidative stress and nitric oxide bioavailability, Fetterman used DHC and DAF2DA respectively, and we also did the same. The e-cigarettes that we used were Aspire Zilos 3, bought from Italian dealers. We applied 2 widely used settings, regular setting and suburb setting. The e-cigarettes were vaped by the Borvald LM4E vaping machine, following the Coresta reference method number 81. The aerosol was bubbled into an e-pitcher filled with 30 ml of trapping solution. The cells used were HAEC, normal primary human aortic endothelial cells, and they were exposed to the aqueous extract for 24 hours for an NRU assay and 90 minutes for the evaluation of the oxidative stress and nitric oxide bioavailability. And now the results. Cytotoxicity assessed by NRU using the regular setting. On the left, figure A, the data are normalized as percentage of control medium. We can see a significant decrease of viability after exposure to 100 millimolar of vehicle control, PGVG and PGVG with vanillin compared to control medium. On the right, figure B, the data are normalized as percentage of the corresponding vehicle control concentration. No significant differences were observed between each concentration of both PGVG and PGVG with vanillin and the corresponding vehicle control concentration. NRU using the suburb setting. On the left, we can see that there is a significant decrease of cells viability after exposure to 100 millimolar and 50 millimolar of vehicle control PGVG and PGVG with vanillin, but also at 25 millimolar of PGVG with vanillin compared to control medium. On the right, again, The LRU data normalization to vehicle control indicated no significant differences in cell viability among PGVG and PGVG with vanillin compared to the vehicle control at its concentration. These results demonstrate that the cytotoxic effect on the cells is predominantly due to the ethanol presence in the vehicle control. In fact, normalization of the data to the respective ethanol concentrations reveals no cytotoxic effect of the aerosols with and without vanillin. The assessment of oxidative stress for the regular setting revealed no significant difference between the different conditions tested in comparison to the medium and vehicle control. However, there is high variability of the results, probably caused by the presence of ethanol. Similar results were observed assessing the suburb setting. Nitric oxide bioavailability. No significant differences were observed between PGVG and PGVG with vanillin and the respective vehicle control concentrations for both regular and suburb settings, indicating that nitric oxide bioavailability was not impaired by vanillin. In conclusion, our experiments revealed some disparities in the results compared to the original study. Many of the effects attributed to vanillin by Fetterman in our work appear to be related to the extensive use of ethanol that they made in their experiments, the use of really liquid formulation, and the standardized exposure method closer to real vaping conditions emerged as critical factors contributing to the observed variations. And with a photo of this year's replica group meeting, I want to thank you for your attention.